Hi, everyone, and welcome to Research Like a Pro with DNA. Today, for our question and answer series video, the topic is how do you add multiple test takers' shared DNA to a Lucidchart diagram? Descendancy diagrams are very helpful to see the descent from the common ancestral couple, and the asterisk is there to remind us that this is the main purpose of making a diagram. However, there are some additional things that I really like about descent diagrams in Lucidchart or whatever program you use. And those are one of them that you can see all the evidence in one place. And in order to see that evidence, you need to add the amount of shared DNA with your test takers so that you can see how much is shared and their descent from the common ancestor all in one place. You can also use your diagrams in written conclusions, whether it be a research report or a proof argument or some other form of conclusion, whether it's a presentation or an email report. So here in this diagram, you can see that I have the common ancestor Cora May Lindsay at the top and the test takers in blue, whose initials are there, and then two DNA matches who descend from different descent lines from Cora. And match 2 was at MyHeritage, so it's in orange. And match 1 was at Ancestry, so it's in green. And that's a common way that I like to color code my diagrams. And you can see that the amount of shared DNA is put there with two of the test takers, and their initials are used to differentiate. But one of the things that a lot of people notice when doing this is that if you have more than just two test takers, there isn't space to keep track of all of the amounts of shared DNA with more than two test takers within that matches box, unless you make the box a non-standard size, which I don't like to do because I do like my boxes to be consistently sized to help with keeping them lined up in the generations. So what to do? There are some different solutions that have been used. So let me share them and then you can decide what works best for your particular project. So if you have multiple test takers, one of the first options that comes to mind is the McGuire chart. And before I dive into what the McGuire chart is, let me just define test takers. When I use the phrase test taker, I'm referring to the fact that you have access to view a DNA test taker's results. So for me, I never use my own personal results. I always use different test takers that are closer generationally, like my grandparents or my mom or a first cousin once removed closer to this research subject. So, whichever test takers you're using, you'll want to chart the matches to the ones that are closest generationally and focus on them. So for this particular project where you can see the examples, there were 4 test takers who descended from Cora May Lindsay, and they're in the blue boxes. Well, a McGuire chart can help you to track the amounts of shared DNA with multiple test takers. And this is what a McGuire chart looks like. This is a very abbreviated, simple one with only 2 matches, but you can see the lines from the test takers here go over and show you how much DNA is shared with that match instead of putting it all in the box. Another thing you can do is use initials, and you can also just keep track of the amounts of shared DNA only in your Airtable base and not in your diagram. And then you can take that information and put it into a table in your written conclusion. And in your written conclusion, you'll want to have at least a basic diagram showing the descent from the common ancestor and a table showing the amounts of shared DNA. So if you think about your end goal, which is probably to have some sort of written conclusion, you won't necessarily need to put a McGuire chart in that conclusion, especially because they're usually too large to fit on a page. And what will usually work better is just having a basic diagram without amounts of shared DNA and then have a table with the shared DNA. Now let's look at each of these in a little more depth. So the McGuire chart is a really great option as you're researching to look at how much DNA is shared between your test takers and the matches. So if I had more matches descending from all of these lines, I would put them over to the right of the diagram. And usually a McGuire chart keeps the test takers to the left so that they can all be showing how much they share as you move their lines along to the right of the other matches, showing how much they share with each descent line. So let's walk through how I made this. So with the DNA match that descends from Amy Mae Ashton, match 1 at Ancestry. And as you can see in the diagram, key green color indicates Ancestry DNA match. Then I checked the amounts of shared DNA with each of these 4 test takers with that match. And then I added a circle shape, which I then condensed down to make that smaller when I was all done. Otherwise, these would be really big circles. So I started off with big circles, then I made them smaller. You can choose however you want. And you can also add more information here, such as the relationship. So that's a common thing to do in a McGuire chart, is to add the relationship between this test taker and this match. The relationship for these test takers and these matches are all the same. They're second cousins. So I didn't add that here. So then after I added the circle for this, I drew a line between the square and the circle and placed it beneath the match where that's the amount of shared DNA. Then drew another line below match 2. And because I don't have this test taker at MyHeritage, I put a question mark there, but I could transfer his results and then check the amount, so I could do that later. Then the other test takers, I repeated the process. So one thing that's nice about this, and let me zoom in, is that you can see, how did I zoom? Okay, there we go. You can see the amounts of DNA that all of these siblings share with match 1 and the variation. So one shares a low amount, 133, and another shares a higher amount, 221, but they are all within what you would expect to see for second cousins. And then you can also see a range here. So this is an advantage of a Lucidchart diagram that incorporates kind of this McGuire chart. And it helps you to see all your evidence in one place, although you probably wouldn't place this into your written conclusion, usually because they are pretty large. Okay, another way to do this is if you only have 2 or 3 test takers, you can put the initials there and put the amount of shared DNA like that. So JWD means that this test taker shares 131 centimorgans with match 2. And then NS shares 221 centimorgans with match 1. And so that's another way you can use more test takers. Now, as you know, sometimes a test taker will only share with, a match will only share with one of your test takers or 2 of them. But if all 4 of them share with one, this would be limiting. Usually what I will do is just put in the top 2 or just put in 1 and then put the rest of them in my Airtable base to track how much the other test takers share. So if you want to always put in at least one amount in your box, then you can focus on the one who shares the highest, or you could just pick one as your main test taker, and then the others you could put into your base. And then if your main test taker, let's say our main test taker is NS, let's say she doesn't match with another person, but we did find RGD matches with that person, then we can put RGD's initials and the amount of shared DNA, and then any other one else that matches that match could go in the Airtable base. So in the Airtable base, you can see the test taker NS and match 1 and how much they share, and then the other test takers with match 1 and how much they share. So that's all tracked here. So you don't really have to track it in your diagram, but it is a nice research tool that you can use to help you see all the evidence in one place, because it allows you to see the relationship and how they're related along with the amount of DNA. So sometimes when I'm not wanting to spend a lot of time on my diagram, I will just track how they descend. I won't put anything else in the box other than their name and a color to indicate where they are matching at. And then I'll just put everything else right here in the Airtable base, knowing that this is where I'm going to get the information to put in my research report or written conclusion and be able to copy and paste this into a table in Word. So how can you copy and paste from Airtable to a Word table or any kind of word processor? What I found that works best is to set up your columns in your Airtable table by hiding unneeded fields. There are a lot of fields in the DNA match details table, and you don't need all of those in your report. And then once you have the columns that you want and you've filtered to see just the rows that you want to put into your report, then count how many rows and columns you'll need for your table. Then create a table in Word that have those dimensions and maybe add another row at the top for the column headers. Then you're going to select all of the cells in Airtable and then Control C or Command C in a Mac to copy. Then when you get over to Word, instead of pasting, you have to first select all the cells in Word in that table and then paste into the cells. And when you do that, the cell data goes into the table. Then you'll add headers for each column and you can even add a table title. So I'll show you how that looks. Okay, so I have some hidden fields, but I want to hide even more. You can click on the field header and click hide field, or you can just go to the hidden fields box and toggle on or off whatever fields you don't want. So I'm going to toggle off a lot of them. I just really want a couple fields. I want how much DNA they share, and then some of the information from the shared centiMorgan project about what the average and the mean and the standard deviation are. Now I'm counting all the rows and all the columns. So it looks like 6 by 10. And now I'm going to select them all. And it doesn't take over the header data. So Control C to copy, and 60 cells were copied, 10 by 6. Now I'm going to insert a table in Word that's 10 by 6, and I did 7, and then select those cells, and then Control V to paste. And now all that information just came right over from Airtable. And I can now add the headers, so the test taker or the match, the company, the shared centiMorgans, the number of segments, the relationship, the range, the average, and one standard deviation below the mean, and one standard deviation above the mean. So now I can make this a little smaller to fit. And now I have a table for my written conclusion that has all the information about how many centimorgans are shared between my test takers and match 1 and match 2. So you can merge the cells of the top row and type in table 1, amounts of shared DNA between descendants of Cora, May, Lindsay. And this is showing that they're all second cousins and that it falls within the range that you would expect. So that's how you would do that. And then here's an example of what a table looks like in your written conclusion. This is a proof argument that's found in Research Like a Pro with DNA, our book. And the title of this proof argument is Who was the father of Bathsheba Tharp Dyer? And this is showing the amounts of shared DNA between two groups of descendants. The C group are descendants of the hypothesized father, and the A group are the descendants of Bathsheba. And because I didn't know which woman was the mother, I couldn't put the exact relationship, but it did have some hypothesized relationships there. And here is what the diagram in the written conclusion looks like, showing just the descent from the common ancestor and no match information as far as like how many centimorgans are shared, because that's all shown in the table. So you could look it up and see, okay, C1 shares whatever. C1 isn't even in the table. C2. C2 shares with A9 38 centimorgans, and with the two other test takers 31 and 15 centimorgans. All right, here's another table. This is one where the relationship was known, and the relationship was, for example, a fifth cousin once removed, and the shared centimorgan project range and mean for the relationship was 0 to 80 centimorgans and 21 centimorgans on average. And then the range for one standard deviation above and below the mean was 6 to 36 centimorgans. So you can see how tables and diagrams can be used in your written conclusion in order to show all the information you need to. But as you're working for a research tool, a McGuire chart or a diagram in Lucidchart that shows amounts of DNA shared with multiple test takers can be useful and help you. So just be aware and think about the end result that you're shooting for when you make your decisions about where to put everything and how you want to keep it all organized. And I don't think that you need to put all the amounts of shared DNA into your Lucidchart diagram. It's much easier to track that in Airtable. So just keep all that in mind when you're making your choices for how you want to do it. All right, thanks.